Kit for determination of the DNA fragmentation level in spermatozoa, Sperm Chromatin Dispersion method. Main compositions: pre-coated slides, gel with low melting point, solution A, solution B, rice stain, rice buffer, SCD preservative reagent. Applicable instruments: 2 to 8 Celsius degrees pharmaceutical refrigerator, general optical microscope, low-speed centrifuge, 80 Celsius degrees dried heater or water bath, 37 Celsius degrees constant temperature water bath. Specimen requirements: the sample should be collected after a minimum of 48 hours, but not longer than 7 days, of sexual abstinence, by masturbation or sexual intercourse using special condoms. The semen sample must be complete in collection. Count sperm concentration by sperm counting chamber. Adjust the concentration of liquefied fresh semen into a 5 to 10 times 10 to the power of 6 per millimeter by normal sodium. If the samples can't complete testing, we will take some semen into EP tube and add with 3 times in volume of SCD preservative reagent. Mix thoroughly and store at -20 Celsius degrees. Preparation of reagents: put the tube that contains gel with low melting point in dried heater or incubation bath and incubate it for 20 minutes at 80 Celsius degrees until the gel dissolved completely. Then transfer the tube to a place at 37 Celsius degrees for use. Test methods: place the pre-coated slide in the fridge at 2 to 8 Celsius degrees for 5 minutes. Dispense 60 microliters of semen samples, whose concentration is 5 to 10 to the power of 6 per millimeters, to the tube of dissolved gel. Mix thoroughly. Take out the pre-coated slide from fridge. Then immediately dispense 30 microliters of sperm suspension made by step 1 to the wells of pre-coated slide. Put the slide cover over the wells of pre-coated slide quickly and gently. Don't press the slide cover and not to make any air bubbles. Then make the gel solidified by leaving the slide at 2 to 8 Celsius degrees refrigerator for 5 minutes. Remove the slide cover carefully. Push the slide cover on side gently until the one end of slider cover goes beyond the pre-coated slide a little. Pinch the edges of the slide cover and remove it horizontally and carefully. Introduce the pre-coated slide vertically into a slide barrel containing solution A. Then incubate it at room temperature 20 to 28 Celsius degrees for 7 minutes accurately. Pick the pre-coated slide up. Clear the fluid remained on the back and slide by filter paper. Then introduce the slide vertically in another slide barrel of solution B. Then incubate it for 25 minutes accurately at room temperature 20 to 28 Celsius degrees. Pick the pre-coated slide up. Clear the fluid remained on the back and the slide by filter paper. Horizontally into the tray filled with enough distilled water for 5 minutes. Renew water once or twice during this step. Pick the pre-coated slide up. Clear the fluid remained on the back and slide by filter paper. Then introduce the slide vertically in another slide barrel of 70% ethanol, 90% ethanol, and 100% ethanol accordingly, each for 2 minutes. Dry the pre-coated slide completely in nature. Dispense 15 to 20 drops of the right stain on the pre-coated slide and then dispense 30 to 40 drops on the right's buffer slowly for staining. Blow the mixture softly by bulb pipette pump on the slide, and don't destroy the surface tension made by the staining. 15 minutes later, rinse the slide softly by distilled water. Make the slide air-dried naturally. Observe 500 spermatozoa under the general optical microscope with 40-field lens and count the sperms with fragmented DNA. Observation and calculation.